have movement of molecules across the cell membrane. So we're going to start off by talking about how molecules move across the cell membrane and the function of the cell membrane. So membranes allow for transport. They allow the exchange of materials from the intracellular fluid compartment to the extracellular fluid compartment. Think about things that the cell needs, nutrients, oxygen. Think about waste products the cell is trying to get rid of, carbon dioxide, and other byproducts of cellular metabolism. The membranes are also selective, so some things can get through. We just described why, right? That non-polar center, some things can get through there and some things cannot. And so this helps to separate two distinct environments. Because some things can go through that membrane and some things cannot, this is what makes the intracellular environment different in terms of the charge, in terms of the proportion of ions and so on, versus the extracellular fluid compartment. The selectivity of that membrane creates two distinct environments inside and outside of the cell. Important things that are being transported: oxygen, nutrients, and waste products. These are the things that need to be able to go through the cell membrane so that the cell can get rid of things it doesn't need and it can obtain important things that it needs for functioning. And when we think about nutrients, we think about amino acids, we think about fats, we think about glucose to metabolize and to make ATP and so on. Let's talk about the selective permeability of these membranes. So some molecules are going to get through, whereas some are not, but this is not just on a polarity basis. So nonpolar molecules obviously can get through the membrane. They can easily pass through the center of that lipid bilayer. Some things like oxygen can also easily get through there. CO2 and fatty acids can diffuse right through the membrane. But ions and polar molecules, water especially, cannot go through the membrane. So glucose, proteins, sodium, potassium, chloride, any charged particle needs a carrier or a channel in order to go through the membrane. It cannot immediately diffuse across that membrane, and we just spent quite a bit of time establishing why. So things that can go through, you kind of want to make a note of that: oxygen, CO2, and fat, or polar or non-polar, molecules. So let's move on to objective 2, which is to explain the role of chemical, electrical, and electrochemical driving force in the passive transport of substances across a membrane. And then we're going to distinguish between passive transport and active transport later on. So passive transport is pretty much easy, spontaneous, downhill transport. This is where we have the movement of substances in their natural gradient. So from an area of high concentration to an area of low concentration. It's going to be according to the concentration of that particle. But it's going to be spontaneous. It's going to generate or liberate energy as opposed to require or use energy. And then and that is why we call it a downhill movement or a spontaneous movement. Active transport, on the other hand, is going to require energy. It's not spontaneous. We can refer to it as an uphill movement. And so think about the ball going from the top of the hill down to the bottom as being a passive transport, easily happening, liberating energy, moving in favor of that gradient. And then think about active transport as being trying to get this green ball from the bottom of this slope to the top. Okay, it's going to require some input of energy. It's not going to happen spontaneously, and it's going to be moving against the natural gradient or gravity, so to speak. Okay. Now, the movement of substances across a membrane is going to be under the influence of two forces. These are the forces we're going to spend quite a bit of time describing and understanding: the chemical force and the electrical force. Now, when something moves across a membrane, it is under the influence of both of these forces together collectively. And so we refer to it as being the electrochemical force. But understand that that is really comprised of two separate forces that are acting on those particles. So we're going to look at them simply by kind of separating them and looking at them individually. And then we're going to put them together and see how they influence the movement overall. So passive movement, we said, is down the force, and active movement is going to be against the force. And so we'll describe what down the force and against the force means in respect to the chemical force first, and then we'll talk about the electrical force afterwards. So let's start out with the chemical driving force. Let's describe it a little bit and talk about what it really means. So it's based upon the concentration gradient or the change in concentration. That's what this symbol means, delta C. So the difference in concentration of a particle across a membrane is what influences the chemical driving force. So very loosely here, we're going to talk about a membrane that's separating two compartments, the intracellular and extracellular. Because we have a difference in concentration of that particle, we automatically have a concentration gradient. The gradient was going to be in order to move the particles from their area of higher concentration to their area of lower concentration. And so it's going to essentially push those particles from one side of the membrane to the other side of the membrane, so long as the membrane is permeable to the particle in question. Okay, so that is the essence of the chemical driving force. So let's illustrate that here a little bit. So we said the chemical driving force is based on two factors. The first factor is that it's based upon the concentration gradient. So the chemical driving force is the movement of particles down their concentration gradient. The second thing we want to point out here is that this is a force that moves the particles from high concentration to low concentration. Okay. And so just to kind of illustrate that here, let's draw a cell. And we have two particles in question. So let's label particle A in red. Right, we have particle A at a lower concentration inside the cell, and it's at a much higher concentration outside the cell. Okay, so we've got a higher concentration of particle A outside and a lower concentration of particle A inside. And then we've got particle B. And particle B is at a higher concentration inside the cell. And then a lower concentration outside. Okay, and I'm sure we can already begin to predict which direction each of these particles is going to move. And that is the force that we call the chemical driving force. It's the force that wants to bring these particles to equilibrium. It's the force that wants to balance the concentration on either side. And so it's going to move particles from an area where they're higher in number to an area where they're lower in number in order to bring about that equilibrium. And so naturally, particle B is going to want to move outside of the cell. So this is the chemical force acting on particle B, moving it outside of the cell in order to bring particle B to equilibrium. And then naturally, we will also see a force trying to move particle A into the cell, again, in an attempt to bring particle A to equilibrium, okay? Or to equalize the concentrations on either side of the membrane, right? We want to make a note here that the membrane is permeable to particle A and B. Okay, this is the only way that we can honor the chemical driving force. The membrane has to be permeable to the particle in question. Okay, so we can see that moving from the inside of the cell, particle B is moving down its concentration gradient. And in order to see what that means, let's kind of draw this out, kind of like a graph here. We have particle B inside, it's at a higher concentration. Outside, it's at a lower concentration. And so this is what we mean by it's moving down its concentration gradient. Okay. And then we can also kind of plot particle A here as well. So particle A is at a higher concentration outside and a lower concentration inside. And so it too is moving down its concentration gradient from outside to inside. Okay. All right, any questions on that before we move on? So that is the concentration gradient. It is a movement of particles from high concentration to low concentration from an area where, through a membrane that is permeable to the particle in question. Excuse me, I had a brain fart. Okay, so let's keep going here. Let me clear this. Okay. So that was the chemical driving force. Let's now switch our gears here to talk about the electrical driving force. And I don't foresee we'll get very far, but we can certainly introduce the concept so you can have an idea and we'll build on this on Monday. But the electrical driving force, some important terminology that we want to appreciate before we actually talk about the electrical driving force is what an ion is. An ion is a charged particle. So think about sodium, potassium, calcium, they are all charged particles. They can be positively charged, that makes them a cation. They can be negatively charged, that makes them an anion. We also want to describe what the word membrane potential means. Membrane potential is represented by this large V and then a small m. This is another force. This is the force of the electrical driving force. This is really caused by the separation of charge across a membrane. So if you've got anions inside the cell and cations outside the cell, you have a separation, you have an unequal distribution of charge across that membrane. This separation, this unequal distribution of charge is an energy. It's a force, it has the potential to do work. That's why we call it a membrane potential. So if I have a lot of negatively charged ions inside the cell, a lot of positively charged ions outside the cell, then there's a force, there's a potential to pull the particles from one side of the cell to the other because of that unequal distribution or unequal separation of the charge. That is what we refer to as the membrane potential. Now, the magnitude of the membrane potential, how strong it is, how forceful it is determined in millivolts. And this is referencing the inside of the cell. So let's say we have a cell at rest. The inside of a cell is usually more negative than the outside of the cell. In other words, there are more negative ions or more anions inside the cell and more cations outside the cell. And so when we say that the membrane potential, for example, is -70, that -70 is referencing inside the cell or the intercellular fluid compartment. So -70 means that there are 70, there are 70 negative anions or negative ions inside the cell in terms of referencing the outside of the cell, if that makes any sense, okay? So when we describe the magnitude of the membrane potential, we're really talking about what's happening inside the cell in reference to what's happening outside the cell. Okay, let's pause here for any questions, and then we'll pick this up on Monday. So what questions do we have about this so far? This is a topic that's traditionally a little gray at the beginning, and then we kind of add clarity as we go. So I really want you to think about the big picture over the weekend. So think about these two forces that are acting on particles. They're influencing whether a particle wants to move in or out of a cell. And they're two separate forces, but because they're both acting on the particle, we're going to combine them at the end. So now we're looking at them individually. We talked about the chemical force based on the concentration of particles. This is basically how much of the particle is present. So whether there's a high concentration or a low concentration. So that's one half of the equation. That's one force. The second half of the equation is the electrical force. So what charge is on the particle? What charge is inside the cell? What charge is outside the cell? What does the membrane potential look like? And that is the second part of the equation, which is the electrical force. Okay, let's see what